we're going over our theme of migratory fishes, science tools to inform management and recovery, with a heavy emphasis on salmon. So as of right now, I see that there's 34 of y'all. I would like to tell you guys to fill out this poll that probably is like on the front of your screen. If you can go over to the next slide, just in case you might have exit out of that screen, you can use this QR code to re-access it. So yes, I'd also like to go over some housekeeping for the rest of this webinar. It is open to the public and will be recorded. You might have just heard an alert, so be aware of that. Our presenters today will have 35 minutes to present and answer questions. So roughly 20 minutes to present with 15 minutes to answer questions. We do encourage a lot of participation discussion. Feel free to use the chat. I'd really like it if everybody could introduce themselves with their name and affiliation. Also, if you have questions for the presenters, we highly recommend using the Q&A. That just keeps it a lot more streamlined. Make sure that we answer everybody's question. And then again, please respond to our polls. You'll see a poll on your screen. And if not, feel free to scan this QR code to access it again. And then I'm just going to wait maybe a couple more minutes before passing it along to our next presenter. And then I'd also like to introduce myself. My name is Megan Nguyen. I am a government program analyst in the research funding unit at the Delta Science Program, Delta Stewardship Council. And I'm here today with my program manager, Dylan Stern of the research funding unit. We also have Emma Askia, which is part of our operations engagement team, who has been wonderful in setting up all of our Zoom and making sure this presentation runs really smoothly. And then we have Trisha Lee as our note taker. Thank you, Trisha. And then we also have Ash and Jessica as our note takers for later as well. So I'm going to pass it along to our lead scientist to get this webinar going, and that is Dr. Lisa Marie Windemeyers.
Thank you so much, Megan. I can see the participants climbing, so it's great to see everyone joining in. Good morning, I'm Lisa Marie, and I'm so glad to welcome you today to this 8th and final seminar in the series. Today, we're going to hear from 3 projects that were funded by the Delta Stewardship Council and the Bureau of Reclamation as part of the 2021 Delta Research Awards, all of which addressed critical knowledge gaps outlined in the 2017 to 2021 Science Action Agenda. Please take a moment now to don't be shy, introduce yourself in the chat, and then also provide your name and affiliation. If you have questions for speakers, we would like you to use the Q&A box. So become familiar with the Q&A box if you can. And before we dive into the presentations, I'd like to share a quick background on the Delta Stewardship Council. Let's move to the next slide, please. Thank you. Great. The council was established under the 2009 Delta Reform Act, and the Delta Science Program is specifically charged with providing the best possible scientific information to inform water and environmental decision-making in the Delta. One key way that we do that is by funding, lots of chatting going on here. One key way we do that is by funding scientific research, including through competitive calls like the Delta Research Awards. Our funding efforts are guided by 2 core tools. There's the Science Action Agenda, which is a collaboratively developed roadmap identifying priority research needs for Delta management, and the Delta Science Tracker, which is a web-based inventory of research projects happening across the region. The purpose of today's event is not just to showcase funded science, these are great projects you're going to hear about, but also to make sure that the results and the management implications are clearly shared and accessible. Next slide, please. Thank you. So towards that end, I want to quickly flag a few other council efforts that I'd like to put on your radar, and maybe folks, you and folks in your network might be interested. The 2025-26 Delta Science Fellows have been announced. The council has awarded over $1 million to support 6 graduate students and postdocs. They'll work with research and community mentors on projects that tackle key science questions in the Delta. And the QR code links here are to an info sheet summarizing all the selected Delta Science Fellow projects. Another QR code link you'll see here is for the TEJ paper, or Tribal and Environmental Justice paper, which is now available and was just announced also at the Stewardship Council meeting this month. So developing this TEJ paper was an important step in acknowledging and addressing the concerns of tribes and environmental justice communities in the Delta and Suisun Marsh. This QR code will take you to the full paper. 2 other announcements. One is this Friday, just 2 days from now, the Coastal and Marine Sciences Institute and the Delta Stewardship Council co-host a symposium. So please check out the microplastics symposium that's happening this Friday. It's a great opportunity to engage with regional climate and community science efforts, and registration is required, although it will be broadcast live and also recorded. And then finally, the Science for Communities webpage is launching May 12th. So we'll dive into this more on the next slide, but just know that the new webpage is going live, and please check out these resources and help spread the word. I'm going to transition to Megan to talk a little bit more about Science for Communities.
Thanks, Lisa Marie. I'm super excited about this program we have coming, our cohort for 2025. So for Science for Communities as background, also known as SFC, connects Delta communities and local agencies with scientists and experts to co-create research that addresses local needs. Since its launching in 2022, SFC has supported projects focused on real-world Delta issues like water quality, mercury exposure, flood risk, harmful algal blooms, and air quality impacts in locations like Stockton. So there are a few benefits for community-based organizations or communities to join SFC, which is partnering with Delta subject matter experts, access to additional resources and funding opportunities, sharing your findings on the SFC website, which will be open May 12th, and then as well as presenting your work at the Bay Delta Science Conference, which I believe a lot of us have attended and liked. So for the community-based organizations, we encourage them to submit their interest forms for the next cohort of spring 2025. They will complete a short survey summarizing their project ideas. Participating SFC teams will start their projects this summer and then finish them by fall 2026. And I just would like to direct to the researchers in the audience right now is that if you are interested in being potential partners through with our community-based organizations that fill out our survey, please reach out to us by this email, just like letting us know that you are interested in being, I guess, engaged with for partnership with whoever, completes our survey as a community. All right, thank you. I'm going to turn it back to Lisa Marie.
Oh, you're on mute.
Thanks, Dylan. So you can see now on the screen that we have, this will be our 8th and final seminar in a series. Here's a look at the previous themes that we explored during the summer of 2024. Each of those seminars featured 2 presentations centered on a shared topic. Today we have 3. There will also be a 10-minute break after the second presentation, probably before 11 a.m. The theme of today's seminar is Migratory Fishes, Science Tools to Inform Management and Recovery. These projects support salmon recovery by improving monitoring, understanding migration patterns, guiding inclusive strategic conservation planning. All 3 covered multiple areas and management needs highlighted in the Science Action Agenda of 2017. And we're now into this 2022 version of the Science Action Agenda. The first project by Rene, Henery, and team developed a collaborative science-based strategy to recover declining Central Valley salmonid populations. This research addressed SAA areas 1 through 5. The second project by Rebecca Buchanan and colleagues developed methods to detect and manage predator interference in salmon telemetry data. This research addressed SAA areas 1, 2, and 5. And the third project by Russell Perry and colleagues used genetic data and modeling to better estimate juvenile spring run Chinook salmon migration. This research addressed SAA areas 2, 4, and 5. So next slide, please. Thank you. And so if you're interested in some of the previous presentations, you can watch recordings of past seminars in this series, which include 2 previous salmon-focused seminars that dealt with telemetry, reservoir cold water releases, predation, and climate change. All of them are available on the Council's YouTube channel, which you can access with this QR code. And so next slide, please. Without further ado, let's invite up Rene Henery for his presentation on the project Reorienting to Recovery. So Rene, are you there?
I am. Can you see and hear me?
Yes, can. Thank you.
Fantastic. Good morning, friends. Thank you for having me. I am excited. I feel like it's an exciting privilege to be able to share with you about reorienting to recovery, a really massive effort over 3 years to, as Lisa renamed, develop a salmon recovery strategy for the Central Valley. This project was funded by a lot of entities. You can see all of their logos down there at the bottom of my slide. But you know, the 2 principal entities were the Delta Science Program and the state water contractors, and really can't stress enough how those 2 entities funding created the possibility for something that's never been done before. And this, despite my sort of extended intro here, I'm going to try to clip along so I can have time for questions at the end, because a lot of material to cover in just this short amount of time. So feel free to post questions in the chat as we go, or use the box as y'all were instructed to do, and we'll get to them at the end, hopefully. Okay, let's see if this works. Oh, there we go. So a little overview of the presentation. I'm going to talk a little bit about why we pursued this task, reorienting to recovery. I'm going to talk a little bit about the approach that we took, which, like I said, I think is new, at least in a lot of facets. I'm going to go through the outcomes of our work and highlight some particular accomplishments. And then I'm going to also talk about where we are now and where we're hoping to go. So the bulk of the presentation is about this 3-year arc that was funded in part by the Delta Science Program, but the project is ongoing, and I think the sort of opportunity is really just growing now that we've accomplished this first piece. So all of this emerged from a couple core challenges. The first one being that salmon are doing really poorly in the Central Valley, and that those generally declining trends have accelerated, as I'm sure everybody on the call is well aware. And this is despite millions and millions of dollars of investment, and that is not unique to California. You know, there's some great papers that have come out in the last few years from the Pacific Northwest and the Columbia Basin especially, that are like, you know, we've spent all this money on habitat. Why aren't we seeing return on that investment? And also, we are very fractured as a people. I don't need to highlight a lot of the social division that's going on right now. There is a huge amount of distrust. There's also an approach to managing our landscape and our human processes that is siloed in different regulatory jurisdictions that don't always talk with one another effectively, or that can't always align well to create larger collective outcomes. And in each of those different flavors of siloing, there are a limited suite of voices and few places where voices all come together and where there is a suitable container to facilitate dialogue across interests and values. And in the midst of all that, an opportunity presented itself. The CSAMP, a collaborative entity that was convened to opine on the biological opinions governing exports originally, but that turned into a forum for dialogue and collaboration and cross-pollination across regulatory entities and water interests and NGOs, had been talking about, can we explore, instead of just trying to understand whether or not exports are harming fish, what it would look like if we focused our investments on those things that would be best for salmon recovery. And in the course of that sort of exploration of a pivot on the part of the CSAMP, I was part of a thing called Uncommon Dialogues that some professors from Stanford organized to sort of try to cultivate new flavors of conversation between water interests in California in this case, and met Jennifer Pierre, the GM of the state water contractors, in that context. And we started having a series of conversations during COVID, when there wasn't a lot of other stuff going on, that sort of turned into some unpacking of a lot of the kind of perceived division between the state water contractors and the NGOs. I'll say real and perceived division. So those things, plus some precedent from the Columbia Basin in the form of the Columbia Basin Partnership, a massive 5-year effort where people got together to think about salmon recovery, developed some shared objectives, and also talked about all the other values that related to recovery. And from that, the Columbia Basin Collaborative formed as an entity to try to work together on salmon recovery. And there had also been some progress in existing California forums, specifically the CVPIA and also the CSAMP, using tools for collaborative decision making. And once sort of all these pieces came together, a new flavor of relationship, a new willingness to look beyond just the regulatory constraints to what recovery might look like, precedent from another place, and in California specifically, some practice using structured decision making. It seemed like there might be an opportunity to explore doing something different that was pan-regulatory and inclusive and designed to explore what we could do for salmon recovery if a lot of our different constituent parts were all aimed in the same direction. And so this new approach that we envisioned, as I mentioned, for a non-regulatory, transparent, inclusive process, had 3 core components. 1, developing a broadly supported scientific definition of salmon recovery, a common objective. 2, summarizing and making transparent the other key values that are often affecting, constraining, how we discuss or how we pursue salmon recovery, because those things that we would do in pursuit of salmon recovery affect these other values too. And then trying to develop a salmon recovery scenario that equitably distributed the pain and the gain of achieving salmon recovery across that range of other values. And you know, in doing this, we also were cognizant that we were trying to develop a model for a different way of operating, where the regulatory spaces in which most of the salmon recovery work is sort of motivated and done could be working together, and that which isn't covered by the regulatory spaces that exist, but is necessary for salmon recovery, could be highlighted, identified, and we could develop a sort of uniform plan for achieving all of it, working together through time. That was the concept. And this is sort of the area of focus. So, you know, we set out to do this and to develop a recovery scenario that advanced salmon recovery in a balanced way for all of the Chinook runs and steelhead across the whole Central Valley, and using a 20-year time horizon as our planning time frame. And these are the phases of the process as we set it up and sort of the timeline for it. So the salmon recovery definition process was in 2021, phase 1 we called that. And we also had some, you know, stakeholder engagement and outreach in that period, more stakeholder engagement and outreach in 2022 with a focus on that values transparency, collecting values and making them transparent and including them in the process. And then phase 3, 2023 and 2024 was really about using that shared objective and those values and the group of people that we'd assembled in the process of collecting those as the basis for a structured decision-making process where we could opine on the different pathways to salmon recovery that we had come up with. And, you know, to do this, we assembled a really diverse team. It included some veterans on the NGO and water side of Delta politics, technical experts from the NGO and water side, consultants to support development of the existing salmon models and data science, the structured decision-making process, outreach and coordination. And similarly, we had many different participant engagement groups that matched the different processes. So for phase 1, we had a science advisory team, a technical team of scientists that came together to develop our salmon recovery definition, our non-regulatory salmon recovery definition. For phase 2, we developed a group that we called the Forum, which is basically everyone we could connect with in the Central Valley Coalition of the Willing that was interested in working on salmon recovery or interested in salmon recovery in some way, touch them in some way. And then for phase 3, a subset of the forum, in addition to the forum, formed this SDM working group, which was a diverse group of representing generally the different constituencies that assembled to specifically develop these scenarios and refine them through time. And as a component of the forum and also in parallel, we had a process to support tribal engagement that was an affinity process facilitated by members of the California Indian Environmental Alliance who are also part of our planning team. And we also had a lot of diverse funding. I showed you the logos in the front end, and I already mentioned the Delta Science Program and the state water contractors. We also received funds from Metropolitan Water District, from NOAA, and from the Water Foundation, and a lot of in-kind contributions from the different project management team members. So now I'm going to talk a little bit about the outcomes of the project. The first one and main one I want to highlight is we developed a salmon recovery definition, and that process, I've been in a lot of salmon scientific processes over the last 20 years in the Central Valley, and I think this one was, for me, unprecedented in that there was really no conflict. We used an approach that was consensus-based, where we decided that that which was beyond the agreement of the group was beyond the realm of science, or in a sufficiently broad space of scientific uncertainty that decisions that we would make about it were really decisions about values. Yeah, and a lot of scientists had different experiences of working with each other, as was reported back to us, and that was fantastic. We also, in phase 2, through a bunch of storytelling workshops, collected 563 individual values from our forum members to get a sense of all of those things that are being touched by salmon recovery actions in the Central Valley. But those values really refined to just 18 performance measures for our structured decision-making process, and really it was like 5 things. Like, people were interested in fish in some place, in some amount of time, water in some place, in some amount of time, land in some place, in some amount of time for some use, money related to fish, water, or land, or ecosystem health. And then there were also a few objectives that related to people's experience of process, like wanting to be included, wanting things to be equitable, wanting to feel like their voices were heard. So it was amazing to go through all those 563 values and have dialogue with the people who shared them, and then understand that really people cared about mostly the same things. And in phase 3, we created an initial recovery scenario that still has quite a bit of refinement to go, but that creates many, many, many more fish in the Central Valley, and that generally all of our diverse participants supported the advancement of. And we learned some key things along the way. We learned that we need integration of what we call the 4 H's: habitat, hydrology, harvest, and hatcheries, in order to get to recovery. We learned that recovery is possible. We had a number of participants who were skeptical of participating in the process because they didn't feel like didn't believe the models could show salmon recovery, because so much of the science that underlies them was performed in a system that is already severely impacted. We learned that a lot of the currently planned actions are important, but not sufficient. And we learned that dry year water is really key to achieving salmon recovery. I'll just pass that one in the interest of time. This is, for those of you, because these are science talks, I had to include a graph. And here's just a picture of some of the different scenarios that we modeled, including the one that we landed on. And you can see baselines condition is down there at the bottom. That dotted line is the salmon doubling goal required under CVPIA. And then these are the results from some of the modeled scenarios that the SDM group came up with. And I want to highlight a few of our accomplishments too. So, creating just a broadly supported pan-regulatory scientific definition for salmon recovery that could potentially guide a larger scale, recovery effort in the Central Valley. Demonstrating through life cycle models that recovery is possible. I know I highlighted that already, but I think this can't be understated. It's really hard to, if you don't know where you're going, it's really hard to get there. And there's a lot of work that's happening to recover salmon, but a lot of those actions which we know are good actions, are happening independently and without being stitched together into a plan and associated with a long-term vision, let alone one that's supported by science. So I think we're now positioned to operate differently as a collective. A broadly supported high-level blueprint for achieving recovery, and also a case study in landscape scale collaborative planning that I think is applicable beyond salmon recovery to any of the water related things that we are engaging in California. And also that's applicable beyond California. I have, since the conclusion of this project, been engaged a bunch with the Columbia Basin Collaborative, and it's great because we learned from them, and there's been this reciprocal cross-pollination where now they're looking at our use of structured decision making and of these decision support models to sort of fill in a missing piece that they've had since they came up with their objectives. We also now have a publicly available decision support model that can do all H integration, salmon life cycle model, so that's available for use in any of the existing planning processes. I mentioned the cross-pollination already, and we created in the process the first iteration of some communication tools, Shiny apps to understand model results, and a story map to understand our process. And now we're at this interesting point where we've created this scientifically supported course, 30,000-foot map for salmon recovery, and we need to bring that down kind of into the existing applied world. And that has a few components. One is sort of looking at all the work that is being done already and sort of connecting the actions identified in our plan with actions that are already happening in the real world, quote unquote, and identifying gaps. Continuing to refine our recovery scenarios. So I mentioned, dry year water is a part of it. There's the opportunity to think about how much water do we need? Can we use it in very specific circumstances? Can we manage it intelligently in the course of multiple years of drought where maybe we don't use as much water in the first year, but it's the second year of a drought, we use more water, things like that. Also developing an implementation framework, thinking about how you, given the existing regulatory spaces where this work's being done, sequence work, how you fill in the gaps, how you would fund the missing pieces that aren't covered in an existing regulatory space, and then just more outreach and engagement to share what we've done, to bring more voices into the process, and to just, develop more sort of engagement and comfort with this new kind of form of potentially operating together. And, success will hinge on continued funding. One of the nice things about the funding from the Delta Science Program was that it was not linked to some particular regulatory entity. And so it was easier for people to step into trust in the process, I think. And going forward, I think our hope is that many of the different agencies, regulatory spaces will all support it so that there's kind of like skin in the game for a number of different people, but no one being owns it, no one entity owns it. And, we'd be happy to have more funding from a lot of our previous supporters, some of whom have already committed to funding the next phase, and we're grateful for that. And we have quite a few resources, project reports, the Shiny app, the story map that I mentioned. And there's been some media from the project at the outset. There's some more media that's coming out soon. And if you want to get to our resources and reports page, you can scan the QR code that's on the screen. And that's what I have. I've got a bunch of supplemental slides though for Q&A, so please feel free to ask any questions, and just thanks for the space to share.
Well, thank you for that excellent presentation, Dr. Henery. I am getting a lot of questions sent here, and so I think I'll start with one. I think it's pretty impressive that you had such solid backing on your recovery definition. One question was, can you expand on what recovery means in this context?
Yeah, I can. It's the super cliff notes of where the group landed is leaned heavily on the viable salmonid population criteria. So the recovery definition was broken down in those areas: abundance, productivity, diversity, and spatial structure. In terms of productivity, it was a positive cohort replacement rate and a growing population to the point where the population is constrained by the limitation of the available habitat. In terms of diversity, it was genetic diversity and life history diversity. And the genetic diversity leaned heavily on the same criteria that are used relative to endangered species summarized in the Lindley et al. 2007 paper. Spatial structure was basically, we want to restore all the historic independent populations. And then abundance was, we want sufficient abundance that, again, based on the equation that's provided for delisting in the Lindley et al. Paper, that the population's at low risk of extinction, but that beyond that, abundance was an expression of the other values that were transformed into performance measures in the structured decision-making process. So, those included, as examples, sufficient fish for commercial and recreational harvest, sufficient fish for nitrogen deposition and ecosystem support. But that is then weighed against performance measures related to land that would need to come out of commission to turn into habitat, or water that, could go to agricultural use that's needed to support populations in certain times. So abundance was really sort of a minimum that was then refined through the weighing of these different performance measures. But as you can see from the graph I showed, the abundance that resulted from the scenario that we landed on ended up being, way, way, way beyond anything that we've seen in the Central Valley recently.
Great, thanks for that answer. I'm not seeing anything in the Q&A, so I'll just keep going with the ones I'm getting through chat. But if anybody has anything to add, please put it in the Q&A if you can. So another question, again, a couple of these on this broad and diverse group of community members and stakeholders that you brought to the table here. Could you share some lessons learned by engaging with such a large group? And also, how did the engagement shape the development of the recovery scenarios and performance measures?
Ooh, good questions and big questions. I'll try to be concise. I think the biggest lesson learned for me was that, and maybe this is just a reinforcement of things that I knew, but it was really potent feeling it, and it's sort of like a combined thing, but there's a certain amount of capacity required, economic capacity, emotional capacity, nervous system capacity, to engage in a process of imagining something different, to contemplate change. And the risk associated with contemplation of change, similarly, is not evenly distributed among people by virtue of their capacity, by virtue of what they're challenged with in the moment. So if you're, concerned immediately with like your livelihood in this second, it becomes really hard sometimes to imagine what's happening 5 years from now. And that inequity in the distribution of pressure and capacity across our participants meant that some people were really excited about the idea of envisioning a future scenario, and other people were like, I'm not sure that this is the best use of my time because I have real problems impacting me today. And so there was a different flavor of trust building that is, I was well aware of the needs for trust building as a function of the way that people had engaged with each other in the past and old adversarial relationships and those sorts of things. But there's a different flavor of trust building required when you're inviting someone into a process, and the risk of their participation in that process is greater for them than it is for you. And also, if you invite them into that process and they join you in it, then there's this tremendous feeling of pressure and responsibility because you are holding them in a place that is more vulnerable for them than it is for you. So there is like a power dynamic in creating a space for people to contemplate change that I think wasn't totally apparent to us, but that we really felt once we got into it. And people shared things like, I really want to be engaged in this, but I'm feeling resistant because I've just been part of so many things that haven't worked. And that was another kind of deep lesson, is that there is kind of a wound, a trauma that is the result of the kind of systemic gaslighting from the world operating in a way that doesn't align with the way that we feel on the inside. And that there's the potential in processes like this that try to imagine something different for that trauma to people to get re-traumatized in that way. It's trauma little t, but it's like a big one, and it creates a ton of apathy. And, I think it's something that a lot of people suffer from. So we had a lot of conversations on the planning team about that and about how we tend to people and about giving people extra space because we knew that they were really stretching to be part of the process. So that's best I can do, is not concise despite my best efforts. But that's the gist on the first one. Remind me of the second part of the question, Lisa Marie, if you would.
Yeah, whether the recovery scenarios and the performance measures that you came up with were a direct result of the engagement, whether you thought that.
Yes.
Yeah.
Short answer is yes. And
Yeah.
I would say not only were they a direct result of the engagement, but they were the direct result of the trust building that we did and the way that we tended to people. I was shocked. I was just actually sharing this with some members of the planning team when we got to the place where we were developing these blended scenarios, at how much forethought different members had given to what an effectively blended scenario would look like. We really, I think, developed a lot of coherence in the group so that by the time we got to these blended scenarios, it wasn't like people working transactionally trying to game the scenarios for their best interest. People were transparent about the values that they had, but everybody was really hive-minding what the collective solution would look like. And I thought that they were there was a direct result of the process and the scenario that we came up with to advance, and that there was agreement on advancing it was too.
Yeah, thank you for that, Rene. And there's some more questions about, more visioning type questions about how you see the data being used by decision makers in the Delta. And there's another, question about, this was focused on salmon. Can you envision what it would look like if it was a multi-species approach? And then there's another one about what you learned about balancing ecological, cultural, and economic goals in terms of this recovery. But I know we don't have a lot of time to go into them, but I know those are the questions that people are asking, and you're not going to see them in your thread right now, I don't think. So maybe we can feed them to you for your response in the Q&A.
Absolutely. And that would be yeah, I'm happy to. And also I'm happy to talk more about this with anyone who's interested. Feel free to reach out, as I know are other members of our planning team, and there's a bunch of different advantages that you get from hearing different voices from our diverse planning team. So if you'd love to know more, we'd love to share more. We're trying to, bring people in because this is very much an effort that will only be successful if it's driven by the collective.
Great, thank you for that, Dr. Henery. And I also want to thank the Bureau of Reclamation for co-funding Reorienting to Recovery, as well as other projects within the Delta Research Awards. And I think if there's no other questions that folks are going to pop in there, we should just move forward with the next presentation. Not seeing anything. Okay.
Thanks again.
Thank you so much. Thank you so much. That was great. Okay, let's move to the next one then. I'd like to invite Dr. Rebecca Buchanan up to the podium, virtual podium here, to give her presentation. When I see Rebecca, I'll know we are set. There she is. Okay. Great. Well, so Rebecca, take it away.
All right, thank you very much, Lisa Marie. And I'm thrilled to follow Renee's discussion of the road to recovery and the expanded way of really thinking about how we might get to that recovery. I'm going to be talking about a kind of a more focused aspect of that, which is itself a very focused aspect of monitoring and evaluation. Our project was to develop a standard operating procedure for diagnosing and addressing predator detections in salmon telemetry data. So it's much more focused again. Before I get going, I would like to acknowledge my co-authors. We are a multi-agency group from the University of Washington, the U.S. Geological Survey, and the U.S. Fish and Wildlife Service. If you'd like to learn more about our project, there's a QR code there at the bottom of the screen to our project in the Delta Science Tracker. Though I recognize that I did not give you a whole lot of time to look at that. Funding came through the 2021 Delta Science Awards, and it came from the Bureau of Reclamation and courtesy of the Delta Stewardship Council. So thank you to both organizations. And we also had some additional collaborators, Dalton Hance from the USGS and Cyril Michel from UC Santa Cruz and NOAA Fisheries. So part of salmon recovery, just a small part, but part of it is monitoring the survival and migration of these fish as they move downriver as juveniles, through the rivers, through the delta, through the bays, out to the ocean. One of the dominant methods for studying the migration and survival of juvenile salmon and steelhead in the delta and the Central Valley as a whole has been acoustic telemetry. It's been used for studying habitat use, movement and survival, the effects of environmental and operational conditions on those measures, and also used or suggested for use in adaptive management. Oh, and the map to the right just shows the receiver locations that were used in, or that have been used in the Sacramento studies. This is a winter run Chinook salmon study released in Redding, and as you can see, they're able to monitor them down to the Golden Gate with many receivers that allowed for reach-specific survival and routing in and through the Delta. One of the, or some of the advantages of using telemetry in monitoring is that it provides a high level of information per fish, so you don't need as many fish released as in some other tagging technologies. It has a flexible study design, and after you have implanted the tags and released them, there's reduced handling, but of course, you have to do some surgery in order to implant the tags, so there's that. There have been advances in tag technology in recent years, quite a few advances. Some of those advances are that the tags are getting smaller, and they're also storing more data, although those two don't always go together. The picture on the right, on the bottom right, is just collected images of some new tags under development in recent years at the Pacific Northwest National Lab. These are not all salmon tags. We have a sturgeon tag, we have an eel or lamprey tag, there's a shad tag, there's an injectable tag. The rice, the grains of rice there are shown for scale. So as you can see, some of these are very small. One thing to say about housekeeping here, on most of my slides, I'll have a box that's bordered in blue at the bottom, and that will have the kind of the big ideas of the slide. So if you get lost, you can look there. The big idea here is there's a lot of acoustic telemetry used in the Delta. So acoustic telemetry is a great technology, but there are some challenges. And one of those challenges is in dealing with tag predation. Tag predation is what we are calling the detection of an acoustic telemetry tag after it has been transferred from the study subject to a predator in a predation event. As you can imagine, this has the potential to bias the study results, whether it is estimating survival, migration, or habitat use. The magnitude of the bias will depend on several factors, including the study objectives, where that predation happened in relation to the study area, and the behavior of the predator after it has predated on the study fish and while it has that active tag in its gut. In some cases, you may observe fairly high bias. In other cases, you may not observe much at all. In general, however, the solution in the Delta has been to apply what we call a predator filter. This is a formal process used to identify and remove detections of predated tags from the analysis. And most of the methods that are used in the Delta are based on presumed behavioral differences between the study fish, which are the smolts, and the predators. And I'm really talking about predators that are other fish here. I'm not really focusing on, say, mammals or avian predation. Right, so we want to apply a predator filter, and there are challenges in doing that. There are a variety of methods used to diagnose tag predation in the Delta, so how do you pick among them? It can be a time-consuming aspect of analyzing the telemetry data, which then delays the results and limits its effective use in adaptive management. It can be subjective, and it can also lead to a lack of replicability, which is challenging for combining data sets across different studies or different researchers. So there's a need for recommended practices and methods to implement those recommendations. So this project was to develop a standard operating procedure for diagnosing tag predation with guidance provided on the filtering approach to use, detection metrics to use, and whether and how you might use reference predator data from intentionally tagged predators in diagnosing tag predation. And then we also developed an R package to implement the various filtering approaches. So this project addresses several areas under the Science Action Agenda. Action Area 5, Modernize Monitoring, Data Management and Modeling. Action Area 2, Capitalize on Existing Data through Increasing Science Synthesis. And Action Area 1, Assess the Human Dimensions of Natural Resource Management Decisions. And it really does addresses all of these by facilitating the timely, reproducible, and well-documented analysis of telemetry data in the Central Valley and Delta. We also analyzed quite a lot of data sets from telemetry studies from both Chinook salmon and predatory fish. So our approach has 4 components, and I'm going to walk through each of these briefly. The first one is reviewing the literature and compiling resources. We started out by developing a resource guide and literature review for addressing tag predation in the Central Valley. And we created this document. It includes an annotated bibliography, a glossary. It categorizes existing methods at the time that we wrote it in 2022. So what methods have been used in the Central Valley and also elsewhere, and also what data sets of predator data, telemetry data sets are available that might be used as reference data. Results are categorized by geographic region, the species of the predatory fish, and the type of the predator filter. And you can find that online at the Columbia Basin Research website. I'm going to have a QR code later. Should have had one on this slide, but I will have a QR code at some point. All right, the second component is to analyze telemetry data from predatory fish. So there's lots of ways you could analyze data. Our particular question was to address which predator data sets can be used as reference data in diagnosing tag predation in a given setting, say for a specific data set of telemetry data from smolt tags. So tags that you put into smolts, which predator data should we use in comparison to that? And we were really interested in whether predator data from one setting could be used in a different setting, or in other words, how the behavior that we can observe of the tagged predators in the data set, how that varies or differs between different habitats and different seasons and different water year types. By habitats, we were pretty broad. We were looking at river versus delta versus the bays. And so we had data from 19 different telemetry studies of predators. Most of those data were of striped bass, largemouth bass, and to some extent channel catfish, but there were a lot of other species that had fewer representation or lower representation in those data sets, but they were there. This covered the years 2009 through 2021 and came from a variety of agencies and also UC Davis. The map on the right shows the distribution and density of the detections of these tagged predators that we had available to us. And you can see that the highest density is in the South Delta. We also had some higher densities in the North Delta and in the bays, but we also had detections in the Sacramento up to almost to Redding and in some of the tributaries and out to the coast. Now, one thing to bear in mind is that these densities, the distribution of detections here is not necessarily representing the predator hotspots. It's really reflecting the objectives of the studies that we were taking the data from. We're repurposing these data. Predator species were not necessarily tagged representative to their distribution throughout the entire area. However, we do see that we have quite a lot of data in some places and a wide-ranging distribution there as well. So our focus was on characterizing the differences in behavior represented by 3 metrics: residence time, migration rate, and upstream movements. I'm going to focus on some results from the residence time analysis and focused on the river and delta regions. So residence time is the length of time that a tagged individual spends within the vicinity of a detection station. And so we characterized residence time by water year type, by season, and by species. This is from the river and delta. We combined those because we didn't really have enough data in the rivers to separate them out from the delta. For species, we only had enough data to separate striped bass out from the other species. So striped bass is in purple and the other species are in gold there. And what really stands out here is that in winter, the predicted residence time increases in the drier years. It's shorter in the wetter years. In the spring, the biggest difference is between species, where the striped bass have a lower predicted residence time than the other species. It's not really surprising since the other species are mostly not as migratory as the striped bass. Okay, so this is interesting on its own, but what do we do with it for our purposes for tag predation? Well, again, we wanted to know what data sets of predator data can be used as reference data for a particular smolt tag study in identifying tag predation. So for example, let's say we have a hypothetical smolt tag study that happened in the spring of a dry year. Which data sets of predator data can we use? Well, we think that we could use the what we call concurrent data, which would be those that also were collected in the spring of a dry year. And that's highlighted in the blue box there. But we might be able to also use data from predators from settings that have a comparable predicted residence time, comparable to what we saw in the dry spring. So that would be the settings that overlap with these red boxes, which are determined by the dry spring predictions. So we should be able to also use data from a below normal or dry or critical year in both winter and spring, but we would not want to be using data from a wet year for comparison to smolt tag data from a dry spring. So it's just an example of how we might go about identifying the necessary or the appropriate reference data. Now, this is based only on residence time. Of course, we have other metrics, and we do recommend that researchers consider all three metrics in identifying the suitable reference data from the predators. That can be complicated. So our R package, it helps identify the suitable predator data for use in the tag predation diagnosis. Okay, so the next component was to develop alternative methods to diagnose tag predation. And we developed two methods here, or two types of methods. We actually had 4 methods in total falling under these 2 categories. The first is what we called an expert opinion rule-based filter. This is a set of univariate criteria that a tag must pass, and it's determined by the user by expert opinion. And we can think of this as implementing a series of tests in a decision tree where each tier of the tree represents a detection event in the tag's detection history. So the tag has to pass a certain number of tests or certain number of criteria at each detection event in order to still be considered to be in a live smolt. So you can think of this as kind of a weight of evidence approach. The other primary type of analysis method that we developed uses multivariate analysis or pattern recognition methods. Each of these methods works by grouping tags that are the most alike in their distribution of the observed detection metrics. And the assumption is that one or more of these clusters represent smolts and the others represent predators. So in this example, dendrogram shown here, one group we interpreted as representing smolts and the other as predators. It does require some interpretation after the clustering is done. So it's not completely without some subjectivity, but it is more based on statistical methods than the rule-based filter. We had 3 methods here that we developed: the hierarchical cluster analysis, a Gaussian mixture model, and a robust principal component analysis. All right. So then the 4th component was to develop software to implement those 4 methods and then compare our diagnosis methods and use that to make more refined recommendations. So developing the software. We've developed a draft package for R. This is the Delta Predator Filter Package, or DPF. It processes telemetry data that is input by the user. It uses a flow line map, which is shown here. And it also uses auxiliary data, such as river discharge from various locations, in particular Freeport and Vernalis. This is shown here for the time period during which we had observations for a particular data set. It computes detection metrics. It identifies the appropriate predator data, implements the diagnosis methods, and visualizes and compares the results. And here's an example of that visualization as an Euler diagram, and I'll come back to that in a minute. Beta testing is ongoing. We've been working on internal beta testing, which we're pretty much done with, and we're just starting on external beta testing. Some of you, I think, maybe volunteered for that, and we'll be reaching out to you shortly. We ourselves have implemented our methods on 9 Chinook data sets, 4 from the Sacramento and North Delta, and 5 from the San Joaquin or South Delta. And we've been implementing a sensitivity analysis. addressing which diagnosis approach should we use, how do they compare, which metrics might we use, how or whether to use predator data in support of the tag diagnosis or tag predation diagnosis, and then there are method-specific questions for each of our 4 methods. So I'm going to just show you some high-level results from one data set. This is the 2017 winter run Chinook salmon. The map that I showed you before of the receiver locations comes from that data, that study. We had 403 juvenile winter run Chinook that were tagged with JSATS tags and released in February of 2017 in Reading. We then implemented all 4 of our tag predation diagnosis methods using a common set of 5 detection history metrics, namely the time since the tag was last seen at a site. This is relevant if the tag is seen multiple times at a given station. Body lengths per second, that's a form of migration rate. Cumulative residence time in a detection range, and also regional residence time, and then the proportion of the time since release that the tag has spent in what we deemed a high risk zone. So we implemented all the methods on this one sample data set and then looked at the results, and I'll show you what those look like in just a moment. One thing to bear in mind, though, as we look at these results is we don't have known fate data. If we did, we would not be trying to develop a tag predation diagnosis methods. So that means that we can't compare our filter output against the truth. All we can do is compare them against each other and look at the effect of our results on the output. So as I mentioned, we used Euler diagrams to compare the output from the different methods. This diagram shows the collection of tags that were flagged as predated by each of the different methods, and of course where those circles overlap, that identifies the tags that were, or the number of tags that had a common outcome from these different methods. So out of the 403 tags that we had, we saw complete agreement on 358 of those tags, or 89%, which is not bad. One tag was flagged by all methods. That's the one that's in all of those circles. So we're pretty certain about that one tag. We think it was probably predated. There are 357 tags that were not flagged by any method. Now that's a lot, but that's not really a concern. It just means that we didn't have evidence of their predation within the detection data itself. So those tags we don't think are going to be potentially biasing our analysis if we were to keep the detections in the data set. But we do have the remaining 45 tags where there is some difference in the outcome from these different methods. And so the conclusion number 1 from this is different methods produce different results. It's pretty obvious looking at this Euler diagram, but it wasn't really obvious that that would be the case until we took a look at it. And we did look at this for all of the 9 data sets that we implemented these on. It's not really surprising that the different methods produce different results because they're keying in on different aspects of the data. So because of that, conclusion 2 is that the best approach may actually use a combination of a rule-based filter and a pattern recognition filter. So perhaps use the rule-based filter to identify the egregious detection histories that are pretty obviously they've been predated, and then perhaps use a pattern recognition filter for the more subtle cases. We're still reviewing that level of recommendation, but we do have some high-level recommendations that are listed here, and we are in the process of refining the other recommendations and putting them into our standard operating procedure document, and they are also implemented in the R package. Our high-level recommendations are, yes, we do think it's important to diagnose or to implement a tag predation diagnosis procedure. We also think it's important that researchers clearly document their diagnosis approach, the metrics that they used, and the criteria used. and also to assess the outcome, perform a sensitivity analysis of the diagnosis decisions. Don't just believe the outcome without examining the results. So these have been listed along with others in our draft SOP, and as I say, we're refining those. We have a couple of weeks left. We'll get that out. And also they've been implemented in the R package. With those two products, we believe that this project contributes to more reproducible analysis, faster study results, which then lead to improved data and science synthesis, improved collaboration, and ideally and eventually improved adaptive management. So I'll wind up by noting scientific contributors. The photos here are the people who were actively engaged in this project. It was a joy to work with all of the folks here. Thanks also to Sarah Michelle and Jeremy Notch from UC Santa Cruz and NOAA Fisheries for the helping us access the predator data. Thanks to the biotelemetry and ITAG work groups for the feedback on previous presentations. And to the beta testers, this is mostly thanks in advance because we're just starting external beta testing on the R package. And a big thanks to the folks who contributed their data, people and agencies. This is fantastic to have access to so much data. And then, of course, thank you to the Bureau of Reclamation and Delta Science program for funding and supporting this project. There is a QR code with a little dinosaur there that will take you to the tag predation website on the Columbia Basin Research website at the University of Washington. Currently, that has the resource guide and literature review if you are interested in that. And soon it will also have our written standard operating procedure document, and it will have the R package in a zip file. They aren't there yet, but they will be shortly. So that is all I have for now. And if we have time, I'll take questions. And like Rene, I also have some extra slides in case anybody wants to see them.
Great. Thank you, Dr. Buchanan. Wow, that was fantastic. And I'm just so excited to see this sort of ensemble approach coming up, looking at all your different approaches. I'm looking forward to seeing that SOP. I'm not seeing questions in the Q&A, which makes me wonder, and there's such a diverse group of people online, if maybe the questions are coming in through chat, I'm not sure. But one of the Q&A questions I did see is in regards to sort of the implications. If you can give an example, you use the 2017 data, for example, but can you give us an example of how the data is showing some guidance? For example, by removing bias, it might give some guidance or ideas about water use decisions or restoration decisions. Is there any input that you can contribute?
Sure. I think in a couple of ways. First, by removing bias in estimation of survival, that would then remove bias in the modeling results that relate survival to operations or, potentially habitat decisions as well. And the other way is by increasing the speed with which the analysis comes out. So in the past, some of these studies have required quite a lengthy analysis, and much of that has been in developing and implementing some sort of predator filter. And different levels of complexity have been used, different approaches across the different studies. And it's hard to compare or combine data from studies that use wildly different predator filters. So this method or this project should help in streamlining that analysis, and whether maybe we won't all be using the same methods, but we'll at least know better which methods people are using, and it shouldn't take 6 months to do a predator analysis. Maybe that's a little long, but it might take a few weeks instead. Or less. You have to process the data, but actually once you have the data processed, the R package works pretty nicely.
Yeah, and following on that question, there's a couple others about the accessibility of this tool, and then any steps that you're doing to encourage adoption of this SOP across different groups, addressing the points you just made.
Yeah, so we have worked with the ITAG and the biotelemetry work group. We've given an earlier version of this presentation to them, and we've engaged with a number of individual researchers who have expressed interest in this, and we're going to be reaching out to them for testing the project or testing the software. And we're also hoping that managers will put a value on this sort of analysis step and a well-documented use of the different tools that we've developed.
Great, thank you. All right, here we have a question from Derek Alcott directly. It says, great presentation. Is it possible to build a large enough, quote, known fate database with the predator tags for aiding in training data? Or is the large tag size of those predator tags a limiting factor in their usefulness?
That is a great question. We haven't yet tried to use the predator tags for this in this project, but I think that would definitely be the next step, to compare the output of the methods that we've developed with the outcome of the predator tags. We'd really like to get our hands on that and try it out. As far as the large tag size, that is an issue, but if we can use similar methods on the predator tag data sets as we're using on the data sets from the smaller tags, that would at least give us some more insight on how our different methods work under a variety of circumstances.
Great. Thank you for that answer. Next question here is from Bob Nyman. Amazing project. Are you getting the same types of results for the Columbia River?
Yeah, another great question. We haven't done this for the Columbia River. We haven't observed the need quite as much in the Columbia River. I think because the telemetry studies, at least that I've been familiar with in the Columbia and the Snake, have been on a larger spatial scale and more in a riverine region where the juvenile fish are expected to be mostly moving in a unidirectional direction, I guess, migration. So that if you see a tag going upstream, it's going to be really more obvious that it's been predated than we might be able to interpret in a complicated region such as the Delta in California. Also, in the Central Valley and Delta studies, the reaches that we are monitoring with the telemetry tags are on a smaller spatial scale than what I've seen in the Columbia. So we haven't developed it for the Columbia, but if we see a need for it, then it would be great to be able to expand it. One of the challenges with using the R package for a different system is that it really depends on the flow line map, which was, of course, developed just for this region. But I think once that, if we can allow for or incorporate a flow line map for a different setting, different system, then the method itself should be transferable.
Hey, Rebecca, this is Russell here. I'll just also add that most of the piscivorous predators in the Columbia are resident, non-migratory kind of predator fish species. And so if juvenile salmon get predated by predators, they're not likely to move long distances across the arrays, whereas in the Central Valley and the Delta, we have to deal with striped bass that are highly mobile and highly migratory, and they're the real kind of critter that causes us problems in the telemetry analyses.
Great.
Yeah, it's a really good point.
Okay, thank you for that. Let's go to the next question from Qingfu Chang. Great and exciting work. I am wondering how the pattern recognition methods associated are associated with a recognized pattern to either smolts or predators?
Yeah, that's a good question. So the pattern recognition methods are really just looking for consistent patterns among different groups of fish within the data set. Or sorry, I shouldn't say fish, I should say tags, because there's a possibility that a tag has been transferred from one fish to another. So tags that have a similar, say, multivariate pattern are going to be grouped in one cluster. Those that have a different multivariate pattern are going to be grouped in another cluster. And I'm being fairly broad here because we do have the different methods that use different approaches to do that. Once that grouping has happened, then it is to some extent up to, well, it is up to the researcher to interpret which group or which cluster represents smolts and which represents predators. And so one way to do that is to project the predator data onto the ordination space of the smolt tag data set and identify where those predator data land. And presumably, ideally, they would mostly land in one or two clusters identified by, and I'm thinking of the robust PCA in this case. And then we could interpret those clusters as probably having been predators, and the other clusters where the predator data do not show up as being smolts. Another possibility is to include the detection data from the known tagged predators in the cluster analysis with the smolts. So perform a clustering procedure on that combined data set, and hopefully the predator data will cluster out, or the predators will cluster out from the smolts, and the clusters that have a majority of the known predators in them, we can assume that any smolt tags that are in those clusters would also be those that were predated, and those clusters that have a minority of the known predators in them are probably actually smolts. So it can be complicated. There's some interpretation there. The R package does include some default approaches to assigning smolts versus predators. So if you don't have predator data, you only have smolt tag data, then it will, apply some default methods. The user is welcome to modify those if they like. You don't have to take the assignment of the predator versus smolt to the different clusters that the package develops. And in fact, we encourage people to look at the clusters and make sure that they make intuitive sense.
Great, thank you. I don't see any more questions being asked, so maybe we'll end on a gaps question that came earlier. Basically, from all of this work you've been doing, are there any gaps in the acoustic telemetry data, whether geographically or temporally, that you've encountered that you in this research that you think we might need to prioritize in the future?
Yeah, that's a great question. Having the tagging data from the known predators was really helpful in identifying which predator reference data can be used from in different regions. However, we didn't have a lot of predator data from the rivers. It was mostly from the delta, and a little bit from the bays, and some from the rivers, but we weren't able to distinguish, say, between the Sacramento River and the San Joaquin River, or among different tributaries, which we had hoped to do. And we didn't have, we had a wide array of species in the predator data, but those data were fairly opportunistic, so it took who they got, and who they got tended to be striped bass and largemouth bass. So it would be good to have, I guess, a wider array of species there. as Derek mentioned, there's the predator tags, which will give you a different signal once it knows it's been predated. And that is, just, it's still, I would say, an evolving technology, but it's starting to be used in the Delta. Expanding that would allow us to do some better testing of these methods as well. And then one of the challenges that we encountered in combining data across multiple data sets was the different densities of the acoustic telemetry arrays and identifying the sensitivity of the different metrics to those different densities. So if you have arrays that are really close together, you're going to have a different perception of, say, going upriver versus downriver than if they are pretty far apart, because they lead to movements, the perception of movements on different spatial scales. I think the more stable array that's been used in the Sacramento over the years has been helpful because it has a more consistent density and placement of arrays.
Great, thank you for that. Okay, well, we're going to move into a break. I just want to make sure folks know what's coming up after our 10-minute break. Thank you again, Dr. Buchanan. The next talk is going to be by Dr. Russell Perry. I think the title you have, Estimating Juvenile Production and Run Timing of Spring Chinook Salmon Leaving the Delta, where he and this team are using genetic data and modeling to better estimate juvenile spring run Chinook salmon migration. So let's come back at 10:36 and lead off talk by Russ Perry. Thank you. Starting to come back. Let's see if I can find Russell Perry online. I'm also very happy to see that we're still at about 80 people. People have stuck online through the break, so that's great. Great, I see you, Dr. Perry. I'm wondering if we can get the slide there. Right. All right, folks are still online. This is great. And I think if you are ready, we can move right to the next speaker, Russell Perry.
Hi, everyone. Let me see if I can. All right. Well, I'm excited to be here today. I'm Russell Perry. I'm a research fish biologist with the Western Fisheries Research Center. And I'm going to talk about estimating juvenile production of spring run Chinook salmon leaving the Delta. And I just want to recognize my co-authors, in particular, Mariah Meek of Michigan State University, who assisted us with some additional genetic sampling, and Brian Piper, who is analyzing that genetic data to estimate assignment rates, and all the other co-authors from many different agencies who have been part of this effort over the years. I'd also like to acknowledge all the support from many different agencies over the year, particularly with the field and tagging efforts that went into the methodologies for generating these estimate abundance I'm going to talk about today, and in particular, the Delta Science Program for supporting this particular effort. Okay, so our project objectives, as the title mentioned, our primary objective is to estimate the abundance of juvenile spring run Chinook salmon leaving the Delta. But in doing that, to identify particular populations, we used genetic techniques, and so that those genetic techniques can have some error in their assigning individuals to particular runs. And so we wanted to also account for error in genetic run assignment. Last, through this process, using historical available data, if we could generate abundance estimates, we also want to provide some guidance and recommendations for improving the sampling methodologies, the sampling designs for improving the abundance estimates. Okay, so why does this research matter? Well, spring run Chinook salmon in the Central Valley were listed as threatened under the Endangered Species Act in 1999, and only a few extant populations remain. More recently, juvenile production estimates, which I'll refer to interchangeably as just abundance of these populations, but juvenile production estimates were recently mandated by the state of California in the incidental take permit. However, juvenile production estimates of spring run Chinook salmon do not currently exist. So this effort is really important for assessing the status of spring run Chinook salmon at different checkpoints in their life history. In terms of stakeholders, I'd say our primary stakeholder is the spring run JPE team. So after the incidental take permit came out, a multi-agency team was put together to develop models and methods and field sampling designs to estimate the abundance of juvenile spring run Chinook salmon. And in particular, they are tasked with estimating abundance of spring run at the entrance to the Delta. And so where this project really fits in and fills a gap is trying to estimate the population abundance as they leave the Delta. In terms of how this fits within the science action agenda, this project capitalizes on existing data through science synthesis. Most of the data already existed, and we're just synthesizing it using modeling techniques and additional analysis. We're modernizing the data management and the modeling by using Bayesian models to integrate many different data sets, from telemetry to codewire tags to trawling catch data to genetic data. And the intent here is to improve understanding between stressors and species and understand more the factors that are affecting spring run Chinook salmon at different points in their life history. Why is estimating abundance of juvenile salmon exiting the Delta important? So I'm showing a map here of the Delta where Chips Island shown with a star there, which is the terminus of the Delta, and and it's important location because this marks the transition from the freshwater environments to the ocean. So if we can estimate abundance there, then we might be able to better understand how water management and environmental factors in freshwater influence, say, survival in that life stage versus the bay and the ocean. This also leverages data from a long-running monitoring program. There's a trawl that operates several days a week at Chips Island to catch juvenile salmon as they emigrate from the Delta, including other fishes. And this monitoring site using a trawl has been operating for over 30 years. And then also, if we have an estimate of abundance of spring run Chinook salmon entering the Delta, and also an estimate exiting the Delta, then we may be able to estimate through Delta survival and understand how the role that the Delta plays in recovery of spring run Chinook salmon. That information can also be used to inform life cycle models, and through that effort to then guide management actions for recovery of spring run. Okay, but there are several major challenges to trying to estimate abundance of populations leaving the Delta. So our first question is, how many fish leave the Delta? I'm showing a picture here of the Chips Island trawl in action. But the trawl only catches, a very small proportion of the fish leaving the Delta. So to estimate abundance, we need to know what proportion of the population the trawl is actually catching. And that's what I'll refer to as trawl efficiency. And estimating abundance at this location in Chips Island is really challenging. So this is showing the channel at Chips Island. It's about 1 kilometer across. and it's strongly tidal. So you may have hundreds of thousands of CFS going upstream on flood tides and downstream on ebb tides. And just to stress how challenging this is, in this Google Earth image actually captured a picture of the trawl. So that little dot in the picture is the trawl in action. So I think it really just kind of stresses how the trawl here is really trying to capture the proverbial needle in the haystack. The second challenge is when the trawl catches individual juvenile salmon, we don't know what population they're coming from. They could be coming from the Sacramento, they could be coming from the San Joaquin. And so we utilize genetic stock identification techniques where we could take a small tissue sample from these juvenile salmon, analyze them, and then assign them to different genetic runs: fall run, late fall run, spring, or winter. As I mentioned earlier on, there can also be assignment error. So we may capture a true spring run, analyze its DNA, and then mistakenly assign it to fall run. So our goal here was also to take several different approaches to assess assignment error. And through this project, we collated all the genetic analyses that had been done for the data collected at the Chips Island. And we were able to compare samples by different labs and use that information to assess whether they agreed or not, and minimize assignment error. That's the approach I'm going to talk about mostly today. Our second approach is using blind test data, where we have a known populations of a sample of fish that we can then apply the genetic techniques to, estimate the assignment errors, and then use that to correct the genetic analysis. We are very far along on that process, and that's the last task in our project, but I'm not really going to focus on that today. Okay, so this study also leverages what I'm going to refer to as this trawl efficiency study, where we've used acoustic telemetry and coded wire tagging data to do paired releases over 8 years, where we were able to estimate the efficiency of the trawl. And that's essentially what allows us to estimate abundance. So this study occurred there's 3 parts to this study. The first is doing paired releases of acoustic tags with a large hundreds of thousands of coded wire tags that are being released along with hatchery releases. So having Rebecca's talk right before this was great, because we're leveraging all of that information that Rebecca talked about to help assist estimating abundance. So the way this works is we release acoustic tags with the coded wire tags. We then have a telemetry array at the trawling site. That telemetry array allows us to estimate the survival of these coded wire tag groups to the trawl site. That then gives us an estimate of how many coded wire tags are available to be able to capture by the trawl, which is the second part of our study here. So given an estimate of survival of coded wire tags to the trawl then captures some of those coded wire tags, and we can use those captures along with the estimate of abundance to estimate the efficiency of the trawl. Last, the trawl is also taking samples, genetic samples of tissues of unmarked fish from fish that are captured at the trawl. Those are genetically analyzed, and we can use those genetic analyses along with the capture probability of the trawl to generate an estimate of abundance for those runs. And we found several different factors that affected trawl efficiency through this study: delta outflow, tides, where the trawl was operating, so what location in the channel, the direction of the trawl. But what we found was the single most important factor was delta outflow. As delta outflow increases, the proportion of fish that the trawl captures decreases significantly, with at the lowest flows, the trawl is capturing only about 1% of fish that are out there, when it's operating. And we were able to use leave one out cross-validation approach to validate the abundance methods. So this shows all the data from those paired release groups. We had 26 paired release groups, over 8,000 acoustic tagged fish released, and 11.4 million coated wire tagged fish released. And we were able to generate a separate estimate of abundance based on the acoustic tags relative to the trawl-based abundance estimates. And what this shows is that the abundance methods were accurate across a wide range of abundances from about 10,000 fish all the way up to about 500,000 fish. Okay, so let's get into the genetics data. This is showing the number of genetic IDs that we have from samples collected at the Chips Island trawl across all years. You'll notice that there are 2 periods where there are a lot more samples collected that are highlighted here, from 2008 to 2011, and then from 2017 through 2021. There was sufficient data here for us to estimate abundance of genetic spring run. I'll also mention we have data from 3 genetics labs: Michigan State University, Oregon State University, which covered the earlier period, and then Kramer Fish Sciences, which is now the lab that is doing analyses from 2016 through today. But also for this period, we could use our abundance model to generate abundance estimates for fish that are classified to run based on their length at date. And so I'm going to compare our genetic spring run abundance estimates to some of these length at date based classifications to run. Okay, so let's talk about assignment error, the first approach that we looked at. Each genetic sample is assigned, has an associated run assignment probability. So there's some probability that a fish belongs to a given run, and then fish are typically assigned to a run using a cutoff value to say the probability is high enough to say that that fish belongs to that run. So by having samples analyzed by different labs, we could then use different cutoff values to see how well these labs agreed. So for example, if we use a 0.5 assignment probability cutoff, what I'm showing here are paired samples where the same fish was analyzed by 2 different labs. And along the diagonal here shows, where in green shows samples that were assigned to the same run by both labs. But the off diagonals show where the labs didn't agree. So highlighted in pink there shows that Oregon State University assigned 135 fish to fall run, but Michigan State University assigned those fish to spring run. But as we varied that cutoff probability, we found that we got better agreement. So in this case, if we use a 0.8 cutoff probability, we had a much broader agreement between these paired samples, which with much fewer disagreements. Although you notice that this reduces the sample size of genetic data that we're able to use for generating abundance estimates. So for what I'm going to show next, we use the 0.8 assignment probability cutoff to minimize any assignment error. And now I'm showing our estimates of abundance, genetic spring run abundance for those 2 periods. So from 2008 through 2011, genetic spring run abundance at Chips Island varied from about 100,000 individuals to about 300,000 individuals. But in this more recent period, we had 2 years that were in that similar range of magnitude, but 3 years where we estimated very low abundance, less than 20,000 juvenile spring run in 2018, 2020, and 2021. Now you may ask, is that a lot? We need some metric to understand, is 300,000 fish, 200,000 fish, is that a lot or a little? Well, now what I'm showing you here is those same genetic spring run estimates relative to the spring run that are assigned based on their length at date. And these for the entire period from 2008 to 2021. So a couple of things to note here is that the length at date abundance estimates are an order of magnitude higher, ranging from roughly 1 million to 5 million compared to the genetic spring run. And you also notice they don't necessarily follow the same pattern. We can't necessarily use these length at date spring run as an index for true spring run. For example, in 2017, you can see that there's a much larger difference between the genetic spring run and the length at date spring run compared to other years. And the genetic, whoops, I went one too fast there. And the genetic composition of those length at date spring run help us understand what's happening here. So it turns out that most fish that are assigned to spring run based on their length are actually genetic fall run, shown in orange here, with the blue being the true genetic spring run. Okay, so in summary, our trawl abundance model, we're able to leverage and synthesize all the historical data that's been collected at Chips Island to estimate abundance of these different runs without bias. We're able to generate genetic-based abundance estimates, as well as abundance estimates of those length at date groups. Our analysis here has shown that we have some very low estimates of abundance in recent years. It also highlights how length at date is a poor index of the true run abundance. I think this is not new, not completely new information. We've known this, but mostly from the context of catch and composition. I think putting it in terms of abundance really helps highlight how genetic stock identification is critical for understanding the status of these populations. And it's also important to consider genetic assignment uncertainty. By utilizing and compiling the genetic data from multiple labs, we were able to minimize the error using the high certainty assignments, but not all the data was used in this analysis. And so the last thing on our to-do list, and we're very far along on this, is to incorporate blind test data where we're able to explicitly quantify misclassification rates. We can then use all of the genetic data to actually apply the misclassification rates and correct the genetic data to estimate abundance. Future steps and actions, we are working to put together the models to estimate spring run abundance annually. We now have the data in hand to be able to do this for 2022 and 2023. We're actively working to transfer the methods, the models, and the code to the Delta Juvenile Fish Monitoring Program to run these models on an annual basis. We're working on publications and data releases to make these abundance estimates available to the public so that they can, and other researchers, so that they can be used and leveraged in things like life cycle models. We have been coordinating with the JPE team, and I am excited to compare our abundance estimates to theirs. They're not far enough along to do that yet, but if we can generate abundance estimates at the entrance to the Delta, we're hoping to be able to look at through Delta survival of these populations. And then there's a new genetic baseline that has been developed and is being used from 2024 onward that has lower assignment error. So we're excited to work with that new genetics information, because the genetic techniques are improving all the time. And with that, I'll take any questions you might have.
Great. Thank you so much, Dr. Perry. Wow, there's so many good questions to follow up with. A lot of them are focused on things that you talked about in your next steps, but let me go to the one that's more about methods. This is coming in from Will Satterthwaite. as you vary the assignment probability cutoff, what biases might you be introducing by excluding fish with, quote, trickier genotypes? For example, is there a correlation between source population and certainty of assignment? For example, my understanding is winter run genotypes stand out very unambiguously.
Yes, that's true. And as a matter of fact, we started our methods with winter run, the abundance methods, because we could basically ignore assignment error because they are assigned with almost 100% probability. I think it can introduce bias. So the main problem with spring run is that there is a small probability of misassigning a fall run to spring run. But that probability is really important because fall run, the abundance of fall run is so high that very small error rates can actually inflate the true spring run abundance, quite significantly. So that's what we're seeing in I think that will come out in the blind test data that we're working with. That's kind of what's apparent right now. So the corrections are, we knew when we were kind of going down the road with spring run that we couldn't ignore that assignment error because it could induce actual biases, even using the genetic data.
Great, thank you. Okay, one more question coming in about methods. With the assays used, this is coming in from Brett Harvey. With the assays used, were you able to identify GREB1L heterozygotes? And maybe
Yes.
define that for me. Thank you.
I'm not the geneticist on this project, but I believe those are the early and late kind of run type. So that might help identify, spring Chinook from, say, fall Chinook because of their migration timing. And that information that's coming mostly from the Kramer Fish Sciences Genetics Lab. And we do have that information for the recent data. I believe they transitioned to including that in in 2020 or 2021. And I'm not sure if they're on the line here, but we have not incorporated that in our analysis, mostly because we have to have the same kind of information across all labs and all those kind of years of data when we did not have that information.
All right, another question gets to broader implications of your genetic modeling approach for other endangered species in the Delta. Could you opine on that?
Could you say it again?
Sure.
Just
Are there broader implications of your genetic modeling approach for other endangered species monitoring efforts in the Delta?
I mean, I think it's really important if you are, the main thing here is like this picture right here, like you don't know what population you're looking at just by catching a fish. So I think if we have, other species where we have, spatially segregated populations that we're interested in managing, and they can be identified using, genetic techniques or, otolith microchemistry is another approach. Then I think combining those genetic identification techniques with sort of, abundance estimation methods kind of basically takes it to the next level for providing information that can directly inform, life cycle models and management. I hope that helps.
Yeah, it's an opining question. And another question was about gaps. You highlighted some in your closing talks, your closing presentation or slides, but also just to help us understand what important data gaps still exist and what would you focus on collecting next to better manage spring run Chinook?
I really think the demographic parameters are, I mentioned survival, right? And we do have acoustic telemetry, like that's, I work a lot on marker capture survival models to understand, how, river flows and water management actions affect survival, but that's not looking at these natural origin populations themselves. So I think if we can really build this architecture to generate, unbiased abundance estimates at different locations in the Delta, that allows us to take it to the next level to then like estimate survival or estimate demographic parameters and understand how they vary through time in response to management actions. And so I think that's sort of the kind of holy grail with this. This abundance estimate is just one piece of that larger kind of demographic picture.
Great, thanks for that clarification. Yeah, and I do think it's a funny comment coming in about Brett Harvey, because there was about 8 Brett Harveys signed up because so many people from there were online. So whomever it was that was actually responsible for the GREB1L question, will the real Brett Harvey please stand up? Okay, I think we are coming to the end. I'm not seeing any more. Actually, here's a new Q&A. Sorry, Darcy Austin. Thank you, Darcy. Have you compared to estimates using probabilistic length at date?
No, but that's a great question. And I would love to incorporate that. So we can, I think as long as I think that's possible. We can estimate using that abundance model. We can estimate abundance of coded tag groups. We can estimate abundance of, unmarked or hatchery fish. As long as we can classify them, we can generate an abundance estimate. So I'd be really interested in working with the JBE team to use different methods of classification, probabilistic or not. And I think we could incorporate that into our abundance model for sure.
Great, good answer. And then one last question, again, coming in from the Brett Harvey group, the team. Are you assuming that survival of acoustic tagged fish is the same as coded wire tagged fish? I'm thinking about tag burden.
Yes, that's an excellent question. And that is the key assumption of those paired releases, is that the acoustic tagged fish have the same survival as the coded wire tagged fish, because we're using that survival estimate to tell us how many coded wire tags are available to be captured. We've looked at that in many different ways to try and assess it, but it is a fundamental assumption, because you cannot estimate survival of the coded wire tags independently. And so as much as we've assessed those assumptions where we can't detect any kind of violations that might indicate there's a difference in survival.
All right, and another question coming in from Bill Poitras. Did you estimate uncertainty around the annual juvenile production estimates?
Yes, there was error bars in those plots that I showed that would be a 90% credible intervals. On average, our abundance models, it's about a 25% coefficient of variation in the abundance estimates generated from those trawl efficiency abundance models.
Great, thank you. And I think that's all the questions we have coming in. There's this closing poll. And I'm going to turn it over to Megan to close us out after saying thank you again, Dr. Russ Perry, for this excellent presentation. Let's turn over to Megan.
I just wanted to thank everybody for being here today, and a special thank you to the presenters for sharing their important research with us. It's been great to see and hear how these research are help bridging their research with you all in the Delta and beyond. I also want to acknowledge Vivian Sue, who's one of our Delta Science State Fellows, who worked really hard in organizing this event. She took the lead on a lot of the behind the scenes work, and her efforts are definitely helped make everything run smoothly. So I also mentioned that, funding research is a core part of our mission at the council, and it's to provide the best available science poor decision-making in the Delta. We know that doing this means creating many spaces for many voices. So thank you again for everyone who joined us, asked questions, and helped make this series a space for learning and connection. So if you missed any of the previous seminars, they are available online on YouTube. So feel free to check in there, as well as all of our info sheets as well. So we hope to see you at future Delta Science program events and just council events in general. Thank you.